Hi student, welcome to Botany class. Today we are going to see the classification of viruses according to David Baltimore. So he can classify the virus based on the RNA or DNA. In previous video we have discussed the classification of virus based on the shape and the symmetry. Now we are going to see the classification of virus according to the David Baltimore. He can classify the virus based on the following criteria. What are the criteria? The presence of whether RNA or DNA. We already know that the virus is contained either RNA or DNA. It's never contained both. Okay. So David Baltimore, he can classify the virus based on the whether it's a RNA virus, RNA containing virus or DNA containing virus. And then the nature of the genome. Nature of the genome, whether it may be RNA or DNA, whether it may be a single standard, that is ssRNA or ssDNA. SS means single standard RNA, single standard DNA, and DS double standard RNA or double standard DNA. Okay, so some of the virus, if it is contained the genetic material is RNA, suppose the RNA virus mean, ribovirus mean, it contained whether the single standard RNA or double standard RNA. They're similar for DNA, deoxyviruses or DNA viruses. It's also maybe contain double standard or single standard. DS mean double standard. SS mean single standard. Okay. Next, the suppose say ssRNA mean it may be a sense or antisense. Suppose SS single standard mean it may be a sense or RNA or antisense RNA. And then the uses of the reverse transcriptase. Okay, so actually the reverse transcriptase enzyme is helpful for suppose say RNA virus. For RNA virus, it must be want to form the protein mean the RNA must be converted into DNA. So from the DNA, the mRNA will be formed. Okay, so the conversion of RNA to DNA with the help of the enzyme reverse transcriptase or RT. Okay, so the uses of reverse transcriptase and mechanism of RNA production. How the RNA production is occurred. So based on these criteria, David Baltimore, he can classify the viruses into 7 classes. Okay, so here we can clearly see that the class 1, class 2, class 3, class 4, 5, 6 and 7 according to David Baltimore. What are the criteria? The genetic material, it may be RNA or DNA, and the genome, it may be a single standard or double standard. Suppose it's a RNA mean either whether it's a antisense or sense RNA. Suppose during the mRNA production, whether it's used a reverse transcriptase and nature of the mRNA production. Based on that, the David Baltimore, he can classify the viruses into 7 classes. So first the viruses, it will be replicate without RT, reverse transcriptase. Suppose it's a DNA virus, mean it do not require reverse transcriptase. Okay, so the replicate with the reverse transcriptase, replicate without reverse transcriptase, replicate with the reverse transcriptase. So the replicate without reverse transcriptase, the viruses contain the 2 genetic material, whether it may be DNA or RNA, that is deoxyviruses or what? Riboviruses. Okay. So the DNA virus, it may be, that is genome nature, it may be double standard or single standard. So the DNA with the double standard without using reverse transcriptase viruses come under the class 1. Once again repeat, the DNA with the double standard or double standard DNA without using reverse transcriptase, these viruses come under the class 1. For class 2, it's a single standard DNA, ssDNA without using reverse transcriptase, they come under the class 2. And then the RNA virus, it will be further divided into 2 types because the nature of the genome is maybe double standard or single standard. Okay, so dsRNA, double stranded RNA, without using reverse transcriptase viruses is come under the class 3. Okay, so suppose it's a single stranded mean it may be a sense or antisense, positive sense and a negative sense. Negative sense is called antisense, positive sense. Okay, so the sense with ssRNA without using reverse transcriptase, this virus come under the class 4. Antisense ssRNA without using reverse transcriptase come under the what? Class 5. Okay, and then the replicate with the reverse transcriptase. So mostly it is a single stranded RNA, that is sense RNA, single stranded sense RNA. So ssRNA with sense, and by using the reverse transcriptase is come under the class 6. And the double stranded dsDNA is using reverse transcriptase. Is using the reverse transcriptase. This virus come under the class 7. Okay, so this is a Baltimore classification. David Baltimore. Next, viral genome. So we already know that the virus is contained any one of the genetic material. It's never contained both. Okay, so and in classification of the Baltimore, we are discussed about the nature of the genome, whether it may be a single stranded or double stranded. It may be linear or circular. Sometimes the DNA it present in the form of a single unit. Sometimes it present in a segmented. Okay, so these are things we are going to see that viral genome. Each virus is contain only one type of nucleic acid, either DNA or RNA. The nucleic acid may be linear or circular. Okay, so linear DNA or linear chromosome, it is a characteristic feature of the eukaryotic, especially for the higher group of organisms. Circular is a characteristic feature of or bacteria. Okay, so the viruses it contain either RNA or DNA. So in the RNA, the linear and circular is present. In the DNA, the linear and circular both are present in both the form, DNA virus and the RNA virus. Okay, so generally the nucleic acid present in a single unit, very common single unit. So sometimes that is wound tumor viruses and the influenza viruses, the DNA found in a various segment, or the nucleic acid present in a number of segment, is not present in a single unit. Okay, it will be present in a number of segment in wound tumor viruses and the influenza viruses. Okay, and then the virus with the DNA is called deoxyviruses, or DNA containing viruses. All the animal viruses are DNA virus or deoxyviruses, except the HIV, not only for HIV, and some of the retroviruses. It is a RNA viruses. Okay, so very common, majority of the animal and bacteria world. It contain which virus? DNA or deoxyvirus, and the viruses with the RNA, that is ribovirus. DNA means deoxy, RNA means ribovirus. Okay, so majority of the plant viruses are RNA virus except the cauliflower mosaic virus. I already told in the previous video. Okay, so cauliflower mosaic virus is a DNA virus. It's a very common. The plant attacking, that is phytophage, is also known as phytophage. Plant attacking virus is also known as phytophage. The phytophage viruses are very common RNA viruses except the cauliflower mosaic virus. Next we have see the viral genome. Okay, once again repeat. So viral genome, it may be DNA mean deoxyviruses, RNA mean riboviruses. In DNA, it may be linear or circular. I already told you. In DNA mean it may be linear or circular. In linear, it may be single stranded or double stranded. We already discussed in the Baltimore classification, no? So David Baltimore, he also said that what? It may be single stranded or double stranded. It may be RNA. The single stranded DNA, double stranded DNA is present the viruses. Single stranded RNA, double stranded RNA is also present the viruses. So DNA means in the linear, it may be single stranded or double stranded. So linear with the single stranded is what? It's a paroviruses. Single stranded or the double stranded DNA with the linear is a herb virus. Okay, and DNA with the circular. It may be a single stranded circular DNA or double stranded circular DNA. Single stranded circular mean M13. Double stranded circular mean is a pagoviruses. Pagoviruses. Okay, RNA. It may be linear or circular. Linear with a single strand, linear with a double strand. Linear with a single strand mean TMV. Linear with a double strand mean reoviruses. Circular with a single strand mean hepatitis D viruses. Okay, and RNA with what? Double strand circular is not exist because RNA is always linear. Very rare case it is a circular, but double strand it will be double stranded circular will be absent in RNA form. Okay, so it is a characteristic features of RNA. In RNA viruses, what? Circular with a double strand is not applicable. Okay, so only what? The single stranded RNA circular with a single stranded, that is the hepatitis D viruses. Okay, so that is this is also example of polymer. What? The DNA with a linear single stranded paraviruses, DNA with a linear with a double stranded herb viruses, circular with a single strand M13, circular with a double strand pagoviruses, RNA linear with a single strand TMV, RNA linear with a double strand reoviruses, RNA with a circular single strand hepatitis D viruses. So we are finished the classification of viruses and the viral genome. Now we are going to see the type study of viruses. So in our textbook they given the two viruses, that is bacteriophage and TMV viruses, tobacco mosaic viruses. Okay, so now tobacco mosaic viruses. So it is one of the plant attacking virus. The genetic material is RNA. Already we know that all the plant attacking viruses are RNA viruses except CMV, cauliflower mosaic virus. Okay, so this virus is first discovered by Dmitry Ivanovsky. Because he is the first man to filter the sap of the infected leaf of tobacco, and by using the bacteria filter, and the filtered juice is sprayed on the healthy plant. So the healthy plant also later suffered by the TMV disease, tobacco mosaic virus disease. Okay, so the tobacco mosaic virus first discovered by Dmitry Ivanovsky in 1892 from a tobacco plant. So this tobacco plant is come under the family Solanaceae. Clear now? Okay. So the vector like the aphids and locusts. So this is mainly carry the disease from the infected plant to healthy plant. Okay, infected plant to healthy plant. The visible symptom in TMV, that is discoloration of the leaf, and then the downward curling and sudden growth. So these are the main symptoms. Okay, discoloration, as such the mottling will be formed. Okay, so the yellow color spot will be appear, and then downward curling in the young leaf, the downward curling and the distraction of the young leaf. The leaf margin, it will be cleaved or it will be teared due to these viruses. Okay. Next is structure of the TMV. In electron microscopic studies, it will be look like rod shaped. So what is the shape of the TMV? That is a rod shaped with help of electron microscopic studies. So it is a helical viruses. Come into the helical symmetry. So that is 300 to 20 nanometer. 300 to into 20 nanometer. Molecular weight of these viruses 39 into 10 to the power of 6 Dalton. 39 into 10 to the power of 6 Dalton. The virion, so virus, the infective nature of the virus, or the viruses are generally called virions. The virion are made up of the two constituents, mainly the center core, that is a genetic material. It will be encircled by, it will be covered by a number of a smaller unit of protein, that is protein subunit. Okay, that is called capsomere. The collection of capsomere is called capsid. Okay, so that is a center core, that is a coiled RNA is present. This coiled RNA, it will be encircled by protein subunit, that is a capsomere. Okay, so its entire structure is called capsid. The outer protecting cover, that is a capsid, that is protein core. Okay, so protein core, that is a capsid, and a core in the center core, that is a nucleic acid. So what is the nucleic acid present in TMV? That is single stranded RNA. Next, structure of TMV. The protein code is made up of 2130 protein subunit. The entire TMV virus is contained 2130 protein subunit. The protein subunit is already we have discussed. That is called capsomere. The protein, the single protein subunit is called capsomere. So here, the single one, that is protein subunit, each is considered a capsomere. Okay. The protein subunit are present around a central single stranded RNA molecule. And the TMV virus contains the genetic material in the center of the TMV. So that is a single stranded RNA. Okay. And then RNA is contain the genetic information necessary for the formation of the complete TMV particle. When the infected plant, that is tobacco plant, inside the tobacco plant, it will be multiplied. How it will multiply? Because it contains all the genetic information for the replication, for increase the population of the TMV virus inside the infected plant. Okay. So the RNA is contain the genetic information necessary for the formation of a complete TMV particle. The RNA is consist of 6500 nucleotide. Okay, so for number of capsomere, that is 2130, and number of nucleotide in the RNA, that is 6500. Next is structure of bacteriophage. So we complete the TMV. Now we are entered to structure of bacteriophage. So it is another example for viruses. So the bacteriophage, while the name itself have the meaning, bacteria killing viruses. Phage means killing. Okay, bacteria means bacteria. Okay, so bacteria attacking viruses. Okay. So the viruses infect the bacteria is called bacteriophage. Literally, it is a eater of bacteria, because at the end of the infection, it will be destroy or it will be kill the bacteria. Okay, so it's a Greek name, that is phage means to eat or to feed, and finally the bacteria will be dead. Okay, so the phage or abundant in, that is the bacteriophage, it's where it is abundantly present mean soil, sewage water, fruit, vegetables and milk. Okay, that is the structure of the bacteriophage. Okay, so it is after the replication, the phage particle will be released from the bacterial cell. It will be killed, or it will rupture the wall of the bacteria. As soon as the bacteria will be died. That is called lysis. So rupture of the wall of the bacteria is called lysis. Okay, so now we are going to structure. That is, it contains the head, tail, and the basal part. Okay, so the T4, especially we are studying the T4 page, is a tadpole cell. T4 page is a tadpole. The young stage of what? Frog. When the frog, it will capture from the egg, it look like what? Tadpole stage. The head will be very broad and the tail will be very narrow. So that is tadpole. It contains a head, collar. So this is a collar, tail, basal plate and the fibers. The entire structure. The head mostly hexagonal. It is made up of the 2000 protein subunit. Okay. So the head region, it is hexagonal in shape. It made up of 2000 protein subunit. Okay. And the long helical tail is consist of the inner tubula. So that is a long helical tail. Its center is the hollow region. The hollow region will be present. That is empty space. Okay, the tubular cover which is connected the head and the tail by the collar. Okay. Next, the basal plate attached to the end of tail. Okay, so basal plate, that is a basal plate, is attached to the tail region. This is a tail region. Okay, it's a collar region, it's a head region. Okay, that is a basal plate. And basal plate is contain 6 spike and the tail fibers. Okay, so that is a 1, 2, 3. Okay, so here 3. So totally 6 tail fiber is present. Okay, 6 spike and 6. So this is a spike. This is a fiber. Okay, so the elongated structure is a fiber, and the very narrow or very small structure is called spike. Okay. So the basal plate contains 6 spike and a tail fiber. These fiber are used to attach the page on the cell wall or the bacteria host during the replication. When it's going to attack the bacteria, so the basal plate is mainly take part, especially for in the basal plate is contained the spike and the tail fiber. So that is very essential for the establishment of attachment with the page particle with the host bacteria. And the double stranded DNA molecule about the 50 millimicron. So it will be 1,000 times larger than the page particle size. It will be compactly packed in the hexagonal head. Okay, one side it contains the double stranded DNA molecule. Is measured about the 50 millimicron. It tightly packed. Why? Because it is the length of the DNA. It will be 1,000 times longer than the page particle. So length of the when compared to length of the page particle, it is 1,000 times larger. So the huge amount of the double stranded DNA molecule is tightly packed in the hexagonal head. Okay. Thank you, student.